Okay, so I am trying this for the second time, as the first time my whole computer froze. So this next section talks about the creation of the protein itself. So the last section that we did talk about, it talked about DNA replication, and that is when your cell is trying to divide. So you need to basically double your DNA so that each cell will have enough DNA to make a brand new cell. So what happens when we need to make a protein? Because DNA is actually stuck inside the nucleus. It can't actually leave. So we have to, our body has adapted in such a way so that we do a bunch of other steps, yes, more steps, in order to be able to make a protein. So, Bio 20, we talked about, or you should have talked about, the fact that we have amino acids in our body. Now, when these amino acids join together, they actually make a protein or a polypeptide. Okay, so the protein would be like your necklace, and the amino acids would be the links joining the necklace together, or the beads of a necklace. Okay, now each protein will be different because it's made up of different amino acids. Now, scientists were able to determine the genetic code of an organism. So we briefly talked about this earlier. So the genetic code is just a specific sequence to identify you as you. So it's a specific sequence of A, T, C's, and G's that make you, you. Now, if you could see me, I'd be doing all these hand signals, and I feel kind of silly because I'm in my office, in my house, just like mumbling away. So, gene expression refers to the actual creation of that protein or trait. So, there are three processes happening within the cell when we talk about genetics. We have replication, we have transcription, and we have translation. Okay, and all three processes are important. You just need to know why they're important. So DNA replication is to replicate the DNA so you can make a new cell and have enough DNA for each cell. Transcription is taking that DNA and converting it into RNA, so that's what you see here, so that the RNA, the code for DNA, can actually leave the cell, or sorry, not leave the cell, leave the nucleus, so that the instruction can code for a specific protein. Now, once the RNA is in the cytoplasm, because that's where it's going, it will translate into a different RNA. Okay, so we have many different types of RNA. The two that you need to know are mRNA and tRNA. There is a bunch more mtRNA, rRNA, but we focus on mRNA, tRNA for Bio 30. So mRNA is just the messenger. So you're taking that mRNA that was created in the nucleus and sending it out into the cytoplasm. In the cytoplasm, you have ribosomes, which you learned in Bio 20. Ribosomes are going to take instructions from the nucleus, which in this case is mRNA, and it will code for a specific protein. So basically, your ribosome is your protein builder, okay? And it will synthesize or create that tRNA or transfer RNA sequence, and this process is known as translation. So transcription, and translation. So this is a very simplistic version of what is happening. Okay, it does go into more detail a bit later, and we'll go over it. So the first thing that we need to learn how to do is read that mRNA sequence. Okay, because we know amino acids join together make up a protein, so we have to figure out what amino acid that mRNA sequence is creating. So DNA comes in a huge 3-meter-ish long sequence, which means that your mRNA could be 3 meters long. So we actually take all of those nitrogen bases. If you think back to the very first video of this section, this chapter, we talked about nitrogen bases and nucleotides. So what we do when we're trying to read a protein is we group them in codons. Okay, now codons is just groupings of 3. Okay, instead of tri, we use the word codon. Okay, now we read the genetic code from the mRNA sequence, not the DNA sequence. And that is super, super important. This is what fudges a lot of students up when it comes to test taking. So you guys have a data booklet. If you brought it home, awesome. If you don't have it at home, I will post a digital version so that you have access to it. I suggest you print it off so you're not flipping back and forth. It's really annoying to flip back and forth. But in your data booklet, you have your mRNA, your messenger RNA codons, and their corresponding amino acid table. Okay, so I tell kids to get out a highlighter and highlight messenger RNA codons right at the top, because then they're like, oh, I'm going to flip to that page on my test. Oh, crap, mRNA. Read from mRNA. Now, when you're reading using this table, okay, so I'm just going to come up with a codon. This is just random. There's nothing special about this one. I'm just putting 3 letters together. So ACU. Now notice that there's U's, not T's, because RNA doesn't have T's, it only has U's. Okay, so we have our first base, we have our second base, we have our third base. Notice this table is organized according to first base, second base, and third base. So you always start at first base, A. Okay, so that means, I'm going to get my highlighter here for a second. I have confined myself to this column. Sorry, this row. I keep messing that up, sorry. So I've confined myself to this row. So A's for first codon, or first first nitrogen base is this row. So now you go to your second base. My second base is C, so second base is up top, so C. Now you've confined yourself to this column. Now you've got to go to a box that has those 2 colors overlapping. So I know that I'm in this box. Now you go to third base, which is U. Third base here, I know I need to stay in this one. So I just follow the U. And I've got to figure out where all 3 are. So ACU is 309. 309? I don't know. Close enough. Okay, so your amino acid is 309. Now, have your data booklet out to the side, and we're going to practice. Your first step is to find your 3 codons. Okay, so I am just going to, so I don't have to keep flipping my pen back and forth, highlight in groups of 3, because those are our codons. That is what we use when we're trying to read from that chart. Okay. Okay, so they're all highlighted. Now I'm going to use my data booklet, so make sure you have it handy. So I'm going to start with AUG. Okay, data booklet, first base, A. Second base, U, and right there, methionine. But notice that there's this little asterisk right here. So down here it says note, AUG is an initiator codon and also codes for the amino acid methionine. Okay, if it is at the beginning, it is acting as your initiator codon or your start codon. Okay, if AUG is in the middle, then you use methionine. But if it's at the beginning, you call it initiator codon or start, whichever one you want to use. I use start because it's easier to write than initiator codon. Okay, now my next 3, CCU. Okay, first base, C. Second base, C. And then U. So that's proline or proline. So when we're writing it out, I am okay if you just put the first 3 letters. So pro. AAG is my next one. So I'm going to go A, and then G. So that's lysine. Welcome to the protein lysine. And I started typing out the word protein at the end of that. Sorry. Oh, I miss teaching in a classroom. So that's 3. So 1, 2, 3. I'm on yellow now. So CGG. So CGG, arginine. So I'm going to put ARG. What does a pirate do? ARG. Okay, now I'm on to purple. CGU. So CG. Oh, look at, I have another arginine. Okay, so you will notice that there are some codons that will code for the same amino acid, and that is totally fine. And we're going to come into start talking about mutations in the next video, and you'll see what that entails and what that can potentially mean. So I'm going to put ARG again. So we have 2 pirates. Okay. Now we have GAU. GAU, aspartate. So I'm going to put ASP. And my last one is UAA. So UAA. Stop. This is a terminator codon. So this just means that it's the end of that amino acid sequence. Okay, so your ribosome will attach where it says start. It'll read until it hits stop, and then the ribosome will detach. Okay, what I want you to do is pause the video and try questions 2 and 3 on your own, and then unpause it to see, if you got it right. Okay, so hopefully you got those 2 correct. Message me if you have any questions. And then the 2 questions is asking, what do you notice about the beginning of the codon, the beginning codon, and what do you notice about the end codon? They are always start and stop. Okay. So the genetic code has 3 important characteristics. So number 1 is redundant. So there is more than 1 code for the same amino acid, as you saw when we were talking about arginine, ARG. It is continuous. So the genetic code is read as continuous. So during translation, it's important to know where that ribosome is stopping and starting to code for a specific protein. And it is universal. So all organisms are built with the same genetic sequences. So A's, T's, C's, G's, and U's. So transcription in detail. So when we are talking about transcription in detail, we are going from DNA into mRNA. Okay, so this is happening inside the nucleus, going into the cytoplasm. So the mRNA will go into the cytoplasm, not the DNA. Okay, so remember how confusing replication was? Transcription is actually quite easy. So DNA is transcribed into mRNA. So the promoter, oops, I need my highlighter back. So the promoter, or the start codon, is the region that will tell the RNA polymerase, so an enzyme, to attach. The RNA polymerase does a variety of things. So it is going to unzip and unwind the DNA strand. Okay, so that was done by helicase in replication. the RNA polymerase is going to add nucleotides to one strand of mRNA. Okay, so that's what your DNA polymerase did. Okay, more specifically, your DNA polymerase 3. So RNA polymerase is doing the job of both helicase and DNA polymerase 3. Okay, so it's going to create one mRNA strand. So T's, okay, so you know how in DNA you had T's and A's flipping and C's and G's flipping. Well, since in mRNA or any RNA there's no T's, you can't just flip A's into T's. Okay, so if you have a T on your DNA sequence, you will have to change it to an A because A's do exist in mRNA, in RNA. A's cannot go to T's, so you have to change it to a U. So when we are looking at this, this side is talking about the DNA strand. So here I'm going to label it so that you know. This would be DNA. I'm going to say original DNA. That looks like DVA, but DNA. And then the other side, this side, is representing your RNA sequence. That's a bad arrow, I'm sorry. Okay. So the, sorry, I need my highlighter back. So as this is happening, it is still moving from 5' to 3' on the new strand, just like in replication. When it reaches the stop codon, the RNA polymerase will detach, and then DNA will zip back up and wind back up. So in the case of transcription, RNA polymerase does it all. Okay, it's a one for all. So this does it all. So it's less complicated and less complicated than replication, because this guy's doing it all instead of having one guy do this, another guy do this, another person do this, and whatnot. Okay, so this big bubble behind is representing your RNA polymerase. Okay, it is unzipping the DNA strand, so you can see the original DNA strand, it was opened or unzipped. Okay, and then we have your RNA, mRNA, being added to create your mRNA sequence. Okay, now translation. This is the one that's a little bit more challenging to understand. So I'm going to link a video after for you to watch what translation and all of this is. I'll try and find a video that explains both transcription and translation. Please, please, please watch to the end of translation because it'll show you exactly what I am trying to explain. It's easier if you see it in a video version because when I'm talking, I'm just talking. If you see a visual, it really helps. So translation. So now this mRNA sequence is going into the cytoplasm. So translation is happening in the cytoplasm. So I'm going to put in cytoplasm. So the T stands for transfer RNA, and it's found in the cytoplasm with an anticodon. Okay. A codon is a group of 3. An anticodon is what would pair with that codon. Okay, so for example, let's get my pen out. If I have AUG, this is my mRNA sequence. Translation, in the process of translation or in tRNA, you have anticodons. So my anticodon for tRNA would be what would bind on the opposite side with this. So A's would normally go to T's, but since we have no T's in RNA, it's a U. So a U, which was normally a T, will get flipped to an A, and G's will go to a C. So the blue is the anticodon, and the green is representing the codon. So you see here, when we're talking about flipping, A's and U's go together, C's and G's go together. Okay? Now, in your anticodon, you actually have the correct amino acid built into this anticodon. So it's kind of like a hat. So imagine this anticodon wearing a hat. On the top of the hat, okay, it's a weird looking hat, but it's a hat, you have the amino acid that corresponds with AUG. So if I look in my data booklet, AUG, if you remember, was this one. Okay, now I don't know if this is at the start of reading or if it's in the middle because I just picked AUG. So I'm going to use methionine. So what happens is methionine is actually attached to this anticodon. So your ribosome is pulling in this whole complex. So this blue and the red, okay? And then it's going to attach all of this red together to make an amino acid sequence, okay? So again, this is very confusing because you can't see the process actually happening. So I tried to write it out in Word format with a picture, but again, please watch the video after of the process as it's occurring. Okay, so two codons of the mRNA will bind to the ribosome. Okay, so if I'm looking here, okay, so here where it started was AUG, which is your start codon. Okay, so the anticodon will be AUG. Methionine used to be attached to this green puppy right here. Okay, now once it moves through this ribosome is moving in this direction. Okay, as it's reading that mRNA sequence. Okay, then it moves to the next, GAA. So free floating in your cytoplasm, you have the anticodon, CUU, so that pairs, so it's like Lego blocks. Okay, GAA, the only Lego block that would match GAA is CUU. Okay, because when you're going from RNA to RNA, C's and G's are going to flip, A's and U's are going to flip. Okay, now the amino acid that corresponds with GAA, if I look here, GAA is glutamate. And if you can see here, glutamate was attached to this anticodon. Okay, now up here, methionine and glutamate are going to attach together. So now we have a dipeptide. Okay, so it's not yet a protein, a big protein. It just has two peptides together to make a dipeptide. Okay, and as soon as you start adding more, it becomes a polypeptide. Now your ribosome is going to chug along, and it's going to find the anticodon for CGC. Well, the opposite of CGC is GCG. GCG, GC, and G codes for here. So if I'm looking, sorry, I was looking up here to try to match this. But again, this is a problem that a lot of students make, and I just made it myself. Okay, messenger RNA. So messenger RNA is down here. So CGC. So we have C, G, C, and that's arginine. So arginine comes in and says, hello, I'm going to attach to the dipeptide to make a polypeptide. Yes, I'm so hilarious. So this arginine will then attach to the glutamate and is going to get pumped out the side. And this will continue to happen as the mRNA sequence is being read. Okay, and elongation will continue until it reaches that stop codon. Okay, so again, watch the video. What we're going to do is a couple questions to try to understand the concept. You will have a summative assignment after this, so please make sure you attempt that. And it will go in PowerSchool 4 marks. Now, anytime you are trying to replicate, translate, transcribe, you are always using your original DNA strand. Okay, so, and that's very important. Always use your original DNA strand when you are transcribing and translating. Okay, so the original DNA strand is as follows. Okay, so I'm going to get out my handy dandy highlighter and start making my codons. Okay, almost done. Okay. Or you noticed in when I did those 2 and 3 up top, I just put a line between them, and that's totally fine too if you want to do that. Okay. So it asks, what would the replicated strand be of this DNA? So now we're going from DNA to DNA. So this doesn't exist. So for DNA to DNA, A's and T's flip, C's and G's flip. Okay, so opposite of A, T, opposite of T, A, opposite of G is C, so TAC. Now we have GCG, so that'll be CGC. Now I flip this, G, because the opposite of C is G, so GAC, TGA, ATA, oh, that's my union, ATA, AGA, and last one, ATC. So that's your replicated strand. Okay, so now it says during transcription, what does the strand change to? Okay, so transcription, we're going from DNA to mRNA. So when we're going from DNA into mRNA, it's important to know what things change to. So A's are going to go to U's, T's are going to go to A's, and then, sorry, I don't know what I was writing there, but C's and G's are going to flip. Okay, so during transcription, what does this strand change to? Always use your original. So you're not going by your replicated, you're going by your original strand, which is this colorful one up top. Okay, so I'm going to change my color here, and I'm just going to make an asterisk for transcription. Look here. Okay, so A's would normally go to T's, but they have to go to a U. Okay, so we have U. T's go to A's, which do exist, and then G's go to C's. G's and C's just flip, so you get CGC. C's change to G's. T's go to A's, and G's go to C's. Now we have ACG, CT. So the A's go to T's, but I don't have T's, so it'll be a U. C's go to G's, T's go to A's. TAT goes to A, T, U, sorry, ATA, but we can't have a T, so AUA. TCT, so we have AGA. And then the last one, we have TAG, so we get A, normally T, but we don't have T's, so we have AUC. Okay, so this is my mRNA sequence. Whoops, mRNA. I don't like that because if you print it off, it's not going to look good. Now it asks, what would your tRNA sequence be? So tRNA, you're going from mRNA to tRNA. Okay, so you're going from an RNA to an RNA. Okay, so this means A's and U's flip, C's and G's flip. So all of these rules apply. Okay, it's a bunch of rules, and it's important that you practice. So now, when you're doing this, you're going to take your mRNA strand and go into tRNA. So I'm looking at this green one to make my blue. So U's flip to A's, A's flip to U's, C, and then C's flip to G's. So G, C, G, C, U, G, A, C, U, U, A, U, U, C, U, and then A's change to U's, U's change to A's, and then C's change to G's. Okay, now the last question says, what is the amino acid sequence to this DNA strand? Okay, so what you need to do, this is going from, I'm going to change to purple, I am going to go mRNA to amino acid table. Okay, so you're using this one. So UAC, UAC, so triazine, TYR. Okay, and these ones, when I made them, I didn't put the start code on. It should have a start code on at the beginning, but I just fudged that when I was making these, so my bad. CGC, CGC, arginine, GAC, so GAC, aspartate, UGA, so UGA, oh, stop. Okay, so normally you would continue reading and then there'd be a stop code on here, but it told me to stop reading at this point. Okay, so your ribosome would actually detach at this point and say, I'm done. Okay, but if you were to continue to code, then you would continue to read on until you reached the actual stop code on here. So this would lead us to talking about mutations. So if this was normally start and this was normally stop, and then you accidentally had a mutation or something, and it caused it to stop here, what effect would that have on the cell? So we'll get into that when we talk about mutations. So pause the video and try this on your own and see if you are correct. Okay, and then I will finish at the bottom. Okay, so hopefully you got this. I put like side notes up here so that you can use it as a guide oops, that's supposed to be a bracket there. So this is given your mRNA sequence. It asks what is your original strand of DNA. So you have to go backwards. So you're going from mRNA into DNA. So C's and G's flip, A's go to T's, U's go to A's. Then it asks what is your tRNA strand. So you can't go in order. You can't use this purple strand anymore. You have to go back to your mRNA because you're only going from mRNA to tRNA. So going from RNA to RNA, A's and U's flip, C's and G's flip. Okay. And then the last one is what is the amino acid sequence, which always comes from your mRNA. So you're going back to this one up top. Okay. And you're reading using this chart to create your amino acid sequence. So hypothesize what would happen if a protein does not have a start or an end codon. Okay. Ribosome does not know where to go. Oops, know where to start reading. Or stop. Okay, so your protein could be super big if it doesn't have an end codon or a stop codon. If it doesn't have a start codon, then you can't even create a protein because the ribosome has no idea where to go. Okay, it's kind of like a seeding plant. So genomics and proteomics. So genomics is the study of genomes. Okay, so the A's, the T's, the C's, and G's, the U's that make you, you. Okay. Proteomics is studying specific proteins in somebody. Okay, so it says in it by a specific genome, so by you, for example. So it's studying the proteins that make you, you. Now, being able to study these two things helps scientists, helps lab technicians, geneticists to be able to specialize medical treatment. As well as potentially to eventually cure cancer, hopefully. So again, if you have any questions, please message me on Remind, and I can either hop onto a Google Meet or I can try to answer your questions through Remind. Please complete the protein synthesis question assignment. It is very, very helpful if you do complete that, not for me, but for you. Okay, especially before your unit exam, which is on May 27th. Okay, good luck.